Our first speaker, Dr. Sudeep Saha. Dr. Sudeep Saha is a biosensor researcher who is working towards developing biosensor devices that enable early diagnosis of disease in order to reduce mortality rate. Mr. Sudeep is currently working as a senior electrical engineer at Biological Dynamics, where he is developing early stage cancer diagnosis system. He has completed PhD in biomedical engineering from McMaster University in Canada. He has worked extensively in optical, electrochemical, and photoelectrochemical biosensor design. There are 2 patent applications pending for his PhD work. Excellent. I cordially welcome Dr. Sudeep Saha for delivering his talk on biosensor for early stage cancer detection.
Thank you for the nice introduction. Are you able to see my screen? Are you able to see the screen?
Yes, you are audible.
Okay, perfect. I'd like to thank you everyone for giving this opportunity to talk about my work. I'm really excited to share my work. This work has been mainly done during my PhD and the postdoc work. So I want to discuss about the early stage cancer diagnosis using electrochemical and photoelectrochemical approach. But I also want to put that into the highlights of Bangladesh and what benefits it can be achieved of doing this early stage diagnosis in Bangladesh. So first thing is like, what is cancer? Cancer is a generic term of a disease. So I just go through a little bit of facts about the cancer. We all know a lot of cancer patients because cancer is a very common disease in Bangladesh and worldwide. So it's a generic term. So I think it's showing up. Okay, so yeah, so it is a large group of diseases that can affect any part of the body. Now, cancer can go to metastasis stage where the cancer doesn't stay in one part of the body, it goes to and it spreads to different parts of the body, and that's called metastasis, which is the later stages of the cancer. The graph you are seeing here is actually showing where Bangladesh stands with the number of cancer patients. It's very high, but it's also not one of the high in the countries, but like among different countries. But you can see that the future prediction, the 77.6% increase in cancer patients from 2020 to 2040. So that's the future prediction, which is very high if you compare with the other countries. Like for example, India is much less, Sri Lanka is also much less, and Myanmar, all our neighboring countries has much less growth rate than Bangladesh, Pakistan, Nepal. Those are like very high risk region. Like India is also very high region, but the growth rate is lower. The increase of cancer and the number of patients in Bangladesh puts into a position that we need to do something towards the cancer for our country. Now in 2020, we can see there's new cases is around 156,000 new cases of cancer and 108,000 deaths happen just for the cancer. Those are some facts for in Bangladesh in 2020, how many patients died from each cancer and what it means in percentage of total death. And you can see that oral cancer, the esophagus cancer, the lung cancer, those are very common cases of cancer in Bangladesh, and there's a lot of people dying every year. And even on the world ranking, the oral and esophagus cancer is number 4, which for a small country like Bangladesh, being number 4 means that we are at a very bad scenario for cancer maintenance or like cancer prevention and treatment what we are doing. And it's right now it's the 6th leading cause of death in Bangladesh, but we need to remember that not all the cases are recorded properly because there's a very poor populations where the cancer doesn't get diagnosed properly. And the other important fact is 66% of our cancer patients in the age range of 30 to 65 years, which means that we are losing our working population. So a big number of our working population is getting lost for the cancer. It's not only the older population that are getting cancer. The working population when it gets cancer is even much bigger economic impact that we are facing. And apart from the economic impact for the cost of the treatment. But you can see that we can prevent this mortality by detecting cancer at the earliest stage. So these are the pie chart that is showing the 5-year mortality versus 5-year survival for like some of the cancers. These are kind of similar for the other cancers as well, but where we say the detected at later stage means when we detect at stage 3 or stage 4. Detected at early stage means stage 1 or 2, and these are from the current data that we have. If we detect at earliest stage before the metastasis happen, like when the cancer is localized, how does it look like? How many patients we are saving now versus like if we detect when it's already started to metastasis, how many patients we can save? So in a lot of cases, we are saving more than 6 to 7 times of the patient. So if here you can see like it's only like 6 to 7% patients we can save, whereas it's almost 70% patients we are saving for lung cancer, oral cancer, ovary cancer, the esophagus cancer. In all the cases, if we can detect this early, we can save a lot of people, and not only saving a lot of people, we're also reducing the cost of treatment. So it is highly important for cancer treatment to detect those cancer early. Now, the first thing, if we want to detect the cancer early, have a biosensor that can detect the cancer. So that's where my work come in. And the biosensor, I would just go through a very brief what the biosensor is. It's a device that consists of a biological recognition system and a transducer. So a biological recognition system, it can be any bioreceptor that like nucleic acid, enzyme, antibody, DNA, RNA, those could be a biorecognition element. And the transducer could be different types: colorimetric, optical, magnetic, electrochemical, photoelectrochemical. There's different types of transducer we can use. And it comes after the output. A device needs to have an output where the output will be in this section. It will be like there's sample processor, there's electronics, there's a display where it shows like what's the. My work mainly involves on the transducer side, electrochemical and photoelectrochemical. And I also, like in my current job, I am working on the electronic side as well. And the advantage of these two transducer method is the output is electronics. And now what's the advantage of having an electronic output is it's very cheap to fabricate, and we have the technology, like the technology is very mature to make electronic device. So it's important to have a device which is cheap and which will not cost like thousands of dollars or like lakhs in the Bangladeshi currency for the cancer diagnosis. Now there are before I go into the biosensor system, I would just briefly describe what are the electrochemical biosensors. In the electrochemical biosensors, we need an electrochemical cell where we have a working electrode, there is a counter electrode, where and there is a reference electrode. So the voltage is applied between working and reference electrode, and the current is going to measure between working and counter electrodes. Now, a lot of the time you see this reference and counter electrode could be a single electrode as well, if it's a 2-electrode system versus it's a 3-electrode system. And the advantage is like electronic readout, as I said, the miniaturization is very easy because of the electronic systems, and the high sensitivity and the multiplexing capability as well. And it needs very small analyte volume. The very good example of this electrochemical sensor and its success is the glucometer. like a lot of us, like who have diabetes, or like we know people who have diabetes, we're using glucometer day by day, like we use a lot, and it's very easy to use. It's very cheap as well. So that's one type of electrochemical sensor. There's iStat blood analyzer, which in a lot of lab people use. You just put a drop and in the blood analyzer, and it can give different types of blood component values. So that's also one of the very commonly used electrochemical biosensors. What is a photoelectrochemical biosensors? The only difference in the photoelectrochemical biosensor is the excitation is done optically. So if I go in my previous slide, here, like we are applying a voltage. So the voltage is applied, like you can see in the graph, that voltage is applied like it increase and decrease. So voltage is work as excitation, and you get a current. You probably get a peak of current depending on what you are using. But in the photoelectrochemistry, we're biasing the working electrode, but we are shining light on the working electrode. So when we do that, the light works as a excitation, and we get the current. It's almost like a concept of the solar cell. So we are shining light, which we can get from the solar light, and we can operate this at a very low potential, very low voltage. That's one of the major advantages of using photoelectrochemical biosensor. So again, it inherits obviously the benefits of electrochemical biosensor, because electronic output and the cheap manufacturing and high sensitivity. But the major advantage is it can operate at a lower bias potential, which is very important for some of the analytes, the DNA or the cellular materials that we need to detect. So I'll go with my work. So I have worked with developing a system using electrochemistry of detecting cancer element. I work mainly with the prostate cancer, but it can be translated to other cancers as well, using exosomal microRNA. Now, exosomes are small extracellular vesicles that carry different element with it. So it carries microRNA, it has protein, it has other elements as well. It has other RNA and DNA, other nucleic acids. So in this work, it's basically I was focusing on detecting exosomal microRNA. These RNAs are, as it says, it's microRNA. So those are RNA, but small in size. It's usually 20 or 18 to 25 base pair, not more than that. It's only 18 to 25 bases. Those are microRNAs. The idea is I had a nanostructured electrode, and we put a captured DNA probe, which is similar in complementary sequence of the microRNA that we are detecting, then the surface blocker, and then we put the microRNA which binds to those DNAs, but there are empty spaces. Then it comes the tag, the signaling barcode with which is methylene blue here, methylene blue, which is a similar sequence of the microRNA, but it's a DNA, and it binds with the empty spaces. So the signal is inversely proportional to the microRNA. So if there is more microRNA binding to the capture, there's a less signaling barcode will be binding. If there are less microRNA binding, there will be more signaling barcode that will be binding. And you can see with this, we can like this is the graph. It shows like a different concentration of microRNA, like what are the current we are getting from methylene blue. So when we run this voltage scan, so this is the current we are getting, and it's increasing concentration wise. We are decreasing, and if we put this log linear scale, it almost provides a linear response. Like it's not linear, I would say log linear response of this data. So this is equations that we are using, so which is very promising, and the limit of detection is very low. It's 122 attomolar. So that means that we can detect the cancer at a very early stages if we can detect at a very low level. So we tested that in patient samples. So we had we used human urine samples for the prostate cancer detections. We lysed the human urine samples to get the exosomes, exosomal microRNA and exosomes. So, and we dropped those, and then we measured the signals, and we got good results. So you can see the prostate cancer patients. So patients should have lower current, the healthy should have higher current. There are so we tested on 10 samples and 4 of the patients showed good results, 4 of the healthy showed good results, but there are 1 which did not show the results that we expected. So I'd say the results were 80% sensitivity and 80% specificity. So that's what we result got. But even with the standard like PSA measurement that we do, like it's only 93%, I think, specificity, and there's a lot of places you can improve this. So I think it's a promising results to go forward. And usually we don't detect microRNA with a single microRNA, the cancer patients. It needs to have a pool. So with this platform, we can just change the complementary, like capture DNA sequence, and we can detect other microRNA and multiplex this device. And similar design I put on the photoelectrochemical approach now. Instead of gold electrode, I was using titanium dioxide electrodes, where we have this type of electrodes, which is very porous, and we again put the probe, capture probe, then target. But here, instead of methylene blue, we are using gold nanoparticle for the amplification barcode. And the advantage here that we are getting signal in 2 stages. First, how much target is binding, because the signal will decrease the current, and when the gold will bind, the current will increase when we shine the light. So we have first current decrease and then current increase. So the percentage of current in decrease also means that we are having a binding event, and percentage of current increase also means that we are having a binding event with the gold. So having these 2 enable us to have a differential signaling. So differential signaling gives 2 stages of signals, and it also reduces the noise a lot. So we'll see the advantage later on, but here you can see how the signal looks like. So at each concentrations of the microRNA, we are seeing the signal decrease at the first stage. So the blue is showing the signal decrease for when we are putting the target, and the gray is showing the gold and complementary DNA, how much signal is increasing. So at each stage, the signal is decreasing, and then correspondingly, signal is increasing more and more. If we do a signal change, the differential signal change, then we can again find this log linear approach, whereas non-complementary lies here. So we can see that we can detect up to 1.58 femtomolar that confidently, but compared to electrochemical approach, the advantage is because of this 2-stage signaling, we can also detect the base pair differences. So if there is a 1 base pair difference or 2 base pair difference, that are also detectable. So we also did some experiments, and we showed that we can detect the base pair differences in those. And we ran those. This experiment done on the buffer. We ran these same experiments on the urine as well, and we have seen good LOD, like 1.96 femtomolar limit of detection with the urine. So those works kind of showed that these are the 2 approaches. These are not only the 2 approaches, these are one of the 2 approaches that we can go for detecting the cancer. And I am also working with the device. There are startups companies in which already started to detect the exosomes in the cancer patients, and then there are clinical relevance of those patients. There are clinical study going on that shows the applications that the detecting exosomes can really detect cancer using AI methods. So that's why I want to highlight that, and I want to acknowledge my professor, and I did the internship where this work mainly has been done, Dr. Dirk and my collaborators as well. So in short, that was my presentations. Thank you, and I would welcome any questions if you have.
Excellent, Dr. Sudeep. We truly enjoyed, and without any interruption in this very excellent, conference room, we enjoyed your talk. And the topic is highly interesting and important for Bangladesh, because cancer is the 6th highest cause of death in our country also, and it is a deadly disease worldwide. Now the house is open for question, comments on the fantastic work presented by Dr. Sudeep Saha. Yes, please introduce yourself and then ask the question.
Thank you, Dr. Sudeep. I'm from Dhaka Medical College. The presentation was really fantastic, but I had some questions. On the first slide, you showed that our cancer prevalence is increasing 77% compared to India or other countries. So my question is, what do you think behind the reason of it? Is it due to the lack of awareness in Bangladesh, or late diagnosis, or lack of early screening? What's your suggestion to minimize this expected prevalence?
So I think there are few reasons. So first, I would say this numbers shows in percentage wise, whereas we are starting from 0.16 and India is starting from 1.32. So if we see like where's the number difference is actually less than India, or I'd say it shows percentage wise, maybe like it shows that we are far worse than India. I'm not sure if that's a true statement if we just see percentage wise. Also, but it's concerning obviously for us because number wise India is still going much higher. That's only because of the number of population. Like Bangladesh has much less number of populations than India. So that's one thing I would say that shows the graph like this. But the other reason is like how many we are detecting. So I think this two number will be increased more because like India, the medical systems, the detection systems, a little bit better than what we are doing, specifically not only because technology things. I'm not referring here the technology, but the awareness and the economic conditions. I would also say like a lot of the times like our cancer get undetected for a big for a portion of the people. So that's one of the things, the awareness I would say, and starting to do some testing. For example, the PAP test that's being very common, but it's not being common in Bangladesh yet for the cervical cancer. That is very important. So there are some tests that started to happen in India where like I think we are still a bit behind, which we can start to do.
Excellent.
So those are the contributing factors.
Excellent, Dr. Sudeep, and thank you for a nice question. Are there any questions?
I have one question.
Yeah, please.
Okay.
Please introduce yourself first
Okay.
and then ask the question.
I am Dr. Pintu Sandochil, CSE department from Kuwait. I have just one question that your research article, I think your research topic is excellent, and I think it's time demanding for Bangladesh. So I have just, it's my curiosity that how you collect data sets from Bangladesh, or how your research field mapping in our countryside, or how your research improve our nation or government like this, applicable, which field your research is applicable in our country. Okay, dhanyabad.
Excellent. Dr. Sudeep.
Thank you for the question. So the data collections is mainly, so I just first go how I collect the data. The data collections is mainly from online. There are several sites like WHO has the Bangladesh data. There's cancer organizations which has the Bangladesh population data. Now the second part of the questions, I think like how it would be applicable. I am currently also working in a biotech startup, and then we're trying to like I'm trying to work in different companies where we are developing the technologies that is developing for cancer diagnosis, early cancer diagnosis. And I'm sure like once the technologies are developed, those can go to Bangladesh as well. Those can come to Bangladesh. There's different biotechs. The one that biological dynamics that I'm working, there's GRAIL, there's Exact Science. They are developing like cell-free DNA or exosome-based cancer diagnosis systems, which would be relevant not only here or in Bangladesh and worldwide.
Excellent. And any other question? Dr. Sudeep. I have a specific question regarding your technique, biosensor technique. You use a different biorecognition system, and here you used the exosome. Exosome, and at early stage of cancer, how sensitive this technique is. This is number one question. And the second question is, the CRISPR technology has emerged as a very powerful for disease diagnosis based in, uh, uh, biomolecules like nucleic acid and others. Is it possible to couple this electrochemical technique with the CRISPR to make more smarter technology?
Thanks. That's actually a very interesting question. So the first question, like how the exosomes might be like helpful. I think exosomes are one of the very good candidates of the early stage diagnosis. The reason behind is exosomes involved in the cellular communication. So when the cancer starts to grow, at the very early stage, the communication goes in where the cancer cells has a different communication. So it has been proven that like exosomes is used to communicate between the cells, and where the cancer cells sense different types of exosomes than the other cells. So at the very early stage, and they communicate through the blood, or like for prostate cancer, it's urine. For the other type of cancer, it could be blood or something else. So the big advantage is we can use the liquid biopsy, like taking the blood sample and then measuring some blood, or taking from urine. So that's the big advantage, because you cannot do the cell culture at the early stage. The second question is the CRISPR based. I think CRISPR is a very that's also in a stage like that would be very helpful to diagnose. The advantage from CRISPR to this, one of the advantages is the electrochemistry and those things is you can go for point of care, but like CRISPR could be very specific as well. It depends how which technology develops fast at one point. But combining those, I think there is a good point of combining these two. Like with the CRISPR-based technology, we can isolate those exosomes and then we can detect, because isolation is also a good part of making these biosensors. So we can do that, like somehow to make these sensors to go into the rural areas. That would be the big advantage if we can combine the CRISPR to this type of sensors.
Excellent. Thank you so much, Dr. Sudeep Saha, for an exciting talk and very important talk. I truly enjoyed. I hope all the audience enjoyed your talk, and we are expecting a point of care early diagnosis of cancer by your future endeavors, and if CRISPR could be added, that would be fantastic. And a new paper possibly you have seen published in Cell, that is cancer specific fungi, or that means cancer specific microbiome has already been discovered, and they have been found in the blood as well as serum at even early stage. So please consider to capture those a specific cancer specific microbiome DNA, and it would be fantastic. Thank you so much, and let us offer him a big hand for a fantastic work, and we wish more success in your future endeavor.